Okay, just go up. Brandywine? Yep.
Welcome to Boston, Massachusetts, and the 2018 International Congress of Plant Pathology. Now please welcome the President of the International Congress of Plant Pathology, Rick Bennett.
Welcome to Boston, and welcome to the 11th International Congress of Plant Pathology, and thank you for joining us in this beautiful city. We're pleased to host this historical meeting in the United States for the first time in 45 years. And we're especially pleased to have all of you join us from around the world to share the latest innovations and discoveries as we explore new ways to connect our discipline with the worldwide community of plant health specialists. This Congress has drawn more than 2,400 attendees from 84 countries, representing academia, industry, and government organizations to exchange current knowledge and present the latest scientific advances regarding agriculture, food security, and plant health issues affecting the world's most important food, feed, and fiber crops. Because of your contributions to this Congress, our program contains more than 75 concurrent sessions with over 400 presentations, and there will be more than 1,200 posters on display throughout the next week. There will be other opportunities to learn, share ideas, and network to renew old friendships, establish new relationships, and gather in a variety of social events. I hope you enjoy your time here in Boston and gain the knowledge you came for while experiencing valuable connections with fellow scientists. In addition to our attendance here in Boston, I'd like to extend a special welcome to all of our colleagues from around the globe who are participating virtually through our live stream video. The live stream is available globally to anyone in the plant pathology community. In addition, we have viewing hubs taking place in France, coordinated by the French Phytopathological Society, and in Nigeria, coordinated by the Phytopathology Society of Nigeria. Tomorrow's plenary session will also be live streamed. I'd now like to introduce a special welcome message from the Mayor of Boston, Marty Walsh.
Hi, I'm Mayor Marty Walsh. On behalf of the city, I want to welcome the 2018 International Congress of Plant Pathology to Boston. And congratulations on your 50th anniversary. I know you've got a record turnout this year, and I hope you get a chance to explore our neighborhoods while you're in town, from our innovation hubs to our world-class museums and our beautiful parks. This is a city that's known for science, education, and discovery. And we're proud that you've chosen Boston as a place to discuss the future of plant health in a global economy. Congratulations and best of luck this week.
On behalf of the ICPP 2018 Organizing Committee, I would like to welcome you all to a historic Congress in Boston. Coordinating this Congress has taken many years and countless hours of volunteer work by many dedicated individuals. I extend a heartfelt thanks to you who closely were involved in the planning of this conference. The organizing team and leadership was responsible for taking the idea of organizing this international Congress and leading their respective teams to successfully implement the program you will experience this week. We tackled some difficult decisions and celebrated successes. There were many hours of conference calls and reaching out to networks to contact colleagues from around the globe. Thank you all for your hard work and dedication to seeing this Congress through to fruition. And to the individuals who volunteered on the Planning Committee, this Congress would not have been possible without your time and dedicated efforts in pulling together the valuable experts and contents for success, along with impressive fundraising from partners in industry and plant pathology societies and foundations, government and individuals, plant pathologists. The Finance and Fundraising Committee raised more than $150,000 to support scholarships for ICP in the way of bursary awards. We gave 139 individuals from countries of developing economies bursary awards to participate in this Congress. The Scientific Program Committee and Program Subcommittee dedicated countless hours to seeing the ICPP 2018 program to fruition. These teams reviewed and finalized 75 concurrent sessions and reviewed over 1,600 abstract submissions for oral presentations. Please give these volunteers a round of applause for all of their work. Finally, we would like to share a special thanks for the sponsorship support we have received from various partners and an impressive display of support and commitment to the plant pathology community. And now, I am pleased to introduce the President of the International Society for Plant Pathology, Dr. Greg Johnson. Dr. Johnson began his tenure at the close of ICPP 2013. He is the founder of Horticulture for Development, a consultancy horticulture and plant pathology group. Dr. Johnson specializes in research and development in tropical and subtropical agriculture with an emphasis on horticultural crops, plant pathology, and development cooperation. Please join me in welcoming Dr. Johnson to the stage.
Hello, everyone. Welcome to the 11th International Congress of Plant Pathology and the General Assembly of the ISPP. On behalf of the Society, I pay tribute to our forebears in plant pathology, to the pioneers of the Massachusetts Bay Colony, to the elders past and present of the First Nations who lived and live in what is now the Commonwealth of Massachusetts, including present-day Boston. I'm glad that you could join us to celebrate this 50th anniversary of the Society, which was founded on the 26th of July 1968 in London. There were more than 1,200 delegates who attended that first Congress, very remarkable for the time, and this week we're expecting more than twice that number. Special welcome to those who've travelled from afar and to our councillors who represent the societies which make up ICPP. Together we have more than 26,000 members worldwide. And this week also, let's think about our colleagues who aren't here, especially those whose lives have been disrupted by war and conflict, and also a special hello to those who are watching online. Speaking on plant pathology and world food problems at the first International Congress in 1968, George Harrar, the then president of the Rockefeller Foundation and plant pathologist, stressed the urgency in solving global problems, including overpopulation, war, poverty and disease, as well as ignorance and social and cultural deprivation. His talk was remarkably prescient in terms of solutions and contributions that plant pathology has made in the 50 years since. And we've also made considerable progress in achieving gender equity for our profession. The next few days are going to be a cavalcade of progress in plant pathology and times for celebration. Key progress and plans for ICPP will be considered by our Society Council on Tuesday evening, and a reminder to councillors to come. And those issues have been summarised in the July issue of the ICPP newsletter, which is available online. I won't repeat them here, but I want to reflect on the past term and look to the future. We adopted the theme, Plant Health is Earth's Wealth, for ICPP, recognising that pathogens don't just threaten food security and well-being. They also affect forests, fibre systems, natural ecosystems, biodiversity, environmental harmony, and they impede trade and market access. And also phytopathology research has been a central focus for discovery and development in biotechnology and plant microbial molecular biology. But for our profession, people are the pivotal element. And while in the coming years the Society will maintain its focus on plant disease impacts on food security, we should and will also foster attention to all facets of our profession through our congresses, our subject matter committees, our website and our publications. And in this light, the ICPP Task Force on Global Food Security, which has more than achieved its objectives, will become a commission working in the same way as other subject matter committees of the Society. Looking ahead, we encourage plant pathologists worldwide to be involved in the proposed United Nations International Year of Plant Health. And if you are interested in signing the petition, I think, for this year, there is available down outside the registration desk, a very large banner that people can sign. And I'd like to also announce here that ICPP has initiated a new task force on plant pathology challenges for 2050 to foresight for and encourage involvement of the young in our profession. Watch this space as the new task force emerges and takes forward its mission. In this term, we've encouraged more equitable representation within ICPP, and the Society plans to obtain full ownership of our journal ahead of the next Congress in Lyon in 2023. We've also streamlined membership arrangements for associated societies and strengthened planning for the next Congress to ensure close oversight by the ICPP. During the opening today, we will present the Jacob Eriksson Prize to Pierre DeWitt, and later in the week we will recognise our 2018 ICPP Fellows and present the Fran Fisher Award. The latter honours a life of commitment to connecting plant pathologists around the globe, and the inaugural recipient is Dr. Charlie Delp. The late great champion of science, Stephen Jay Gould of Harvard University, noted, "We can usually tell the history of a profession as a pageant of changing ideas and proponents." And he went on to say that science is and must be culturally embedded. What else could the product of human passion be? So my challenge to you in this Congress in the future is, let's work together to better harness knowledge and passion to improve both the progress of science and the equity of advancement of our societies and our profession. And as Stephen Jay Gould said, despite the wicked curves, detours and delays, the rocky road towards scientific truth is a great trip. I now take great pleasure in introducing Dr. Moritz Ramstedt, who's chair of the Jacob Eriksson Commission, and he will introduce the Jacob Eriksson Prize. Thank you.
Thank you, Dr. Johnson. Established in 1923 at an international conference on phytopathology and economic entomology in the Netherlands, actually in Wageningen, the Jacob Eriksson Prize is to encourage creative studies of plant pathogens and the processes of disease and disease development in plants. It was first awarded in 1930 to Professor John Hubert Craig of the University of Manitoba, Canada. Then a fund was established in the 1930s, with contributions from Professor Quangier from the Netherlands and Professor Yashevsky from Russia, and the American Phytopathological Society. This has been administered by the Royal Swedish Academy since the 1950s, and now it's used to mint the Jacob Eriksson Gold Medal. The first recipient of the medal, awarded in 1959, was Professor Jim Hurst of Rothamsted Experimental Station, England. The prize is named in honor of Jacob Eriksson, that lived between 1848 and 1931, a prominent Swedish mycologist and plant pathologist, who wrote in 4 languages and promoted cooperation between plant pathologists all over the world. Eriksson's writing and ideas were instrumental in the formation of the International Society for Plant Pathology in 1968, at the first International Congress of Plant Pathology. And now I would like to welcome to the ceremony Ulla Jörstrup, representative of the Swedish Council in Boston, who will present the Jacob Eriksson Medal.
So, thank you, Dr. Ramstedt. Now, on advice of the Jacob Eriksson Commission, with endorsement by the International Society for Plant Pathology, the 12th Jacob Eriksson Prize is awarded to Emeritus Professor Pierre de Witt of the Laboratory of Phytopathology, Wageningen University, the Netherlands. Professor de Witt is being recognized for his pioneering research in molecular plant pathology and plant-microbe interactions. I please invite Professor.
So colleagues would now like to invite Pierre de Witt to deliver the 2018 Jacob Eriksson oration, From Elicitors to Effect-Assisted Disease Resistance Breeding. Pierre de Witt.
Thank you, Dr. Johnson. Thank you, representative of the Swedish Consulate in Boston, Ulla Jostrup. Thank you, International Jacob Eriksson Commission. I really feel honored with this award. It's now my task to lead you through this talk, which is called From Effectors to Effector-Assisted Disease-Resistant Breeding. I started my studies in 1968. It's 50 years ago also, so at the same time as the International Society of Plant Pathology was founded. And I was motivated by, the name is already mentioned, Rachel Carson. And I read the book Silent Spring. And Silent Spring is a book which is, in the 60s, motivated the Kennedy administration to look for new regulations on pesticide use. On the right side of this slide, you see a picture of me with the book, and it was given to me as a painting at my farewell party a few years ago. It shows me looking at the golf course, and at the moment I'm very interested in curing diseases on golf courses by ecological ways, so not using pesticides, but cultural methods and so on. And in the frame you see part of my work I did on host-pathogen interactions. In those days, in the 60s and the 70s, everybody was after finding a molecular proof for the famous gene-for-gene hypothesis put forward by Harold Floor. And Harold had found the genetics of the interaction between flax and flax rust, and this is the famous quadratic check. If you look at the upper part of the slide, you see a plant with a resistant gene and a plant that is susceptible, and you see a fungus with an avirulent gene, and below a fungus that is virulent. And you can see that in three occasions the interaction is compatible, or the fungus causes disease, and in one case you have resistance. And the most simple way to explain it was to think in receptors and ligands. And where the ligand would fit the receptor, you would get an induction of defense response, which is exemplified by a hypersensitive response, a type of programmed cell death. When the ligand doesn't fit, you don't get an induction, and that's why you get disease and no hypersensitive response. So when I started, there was a big problem in the Netherlands, but also in those days there was a problem in the US and Canada with leaf mold of tomato. There were many resistant genes available in some cultivars, and there were already quite a few races that could overcome resistance. So if you look on the left side, you see all the resistant genes. CF stands for Cladosporium fulvum resistance. On the upper side, horizontally, you see all the races from 0, 5, 4, 5, and 2, 4, 5. And when there is an R, there's resistance, and when you see an S, there's susceptibility. So it looks like a checkerboard. So the question was, what is the reasoning behind this? Why do some plants respond and some others don't? So if you have a susceptible plant, the plant does not respond and doesn't give a defense response. That's reflected here. You see a spore landing, it enters to a stomates, and then it colonizes the intercellular space, and you don't see cells that do respond. However, if you have a resistant plant, or tomato cultivar, and then a spore lands on this plant, very soon after penetration, the fungus is recognized, and the plant sacrifices a few cells. That's what we call the hypersensitive response, a type of programmed cell death. So the question, what is recognized? And these were the days of elicitors and resistant genes. And in those days, when I was a student, everybody was advocating you should grow the fungus in vitro. Get rid of the plant. Work with a clean culture. The problem was, you see here on top the fungal, the pathogen, and the bottom you see a plant cell. We were able to isolate elicitors from the cell wall. These are the green and the red triangles. And we were able to isolate oligosaccharides, glycopeptides, and chitin, oligochitins. But in all cases, irrespective whether we will isolate those compounds from a virulent or avirulent strain, it didn't matter. In all cases, these compounds did induce a defense response. What you see downstream, the defense genes will be activated. You see that by Chalcon synthase, by phenylammonia lyase. Then you get phytoalexin production. And this was also the era of phytoalexins. Probably some of you remember pyzatin, phaseolin, rishetin, and also pathogen-related proteins. So these were secreted by the host, and as a result of that, the pathogen would be inhibited. That's what you see on top. At the same time, not only locally, but at a distance, systemically, you got induced resistance. But as I concluded here at the end, nobody in the world could find specific compounds. All these compounds were non-specific. And I also acknowledge here my role models on the right side of the slide. Or for you, it's right side. It's Noel Keane, who coined the term elicitor. It's Joe Kuch, where I spent my sabbatical, and it's Peter Albersheim, who was a specialist on oligosaccharides. So in search for specific elicitors, we did grow the fungus on susceptible plants, and because this fungus is growing intercellularly, we could isolate apoplastic fluids, as you see on the left side. We could put the leaves in the centrifuge tube, and then on the bottom of the tube you see this black stuff. That's the apoplastic fluid, and we would then inject into the different cultivars. And to our big surprise, we could see that the responses from different races, you see on the right side, where you see the yellow spots, that is a kind of defense response, the HR. You only see when the cultivar is resistant to that race from which the apoplastic fluid is obtained. This was already done at the end of my PhD. It took ages before we could identify the first compound. But we were lucky in terms of a bioassay. So we could fractionate the apoplastic fluids by using columns, by using electrophoresis, by using mass spectrometry. And then we used the bioassay and the fractions, and at the end, after some years, we ended up with a pure protein. But those were the days before around molecular biology. So we had a protein of 28 amino acids, but we didn't know the amino acid sequence. So we had to go from sequence to messenger RNA to gene. But it took a long time before we could go from the protein to the gene, because, as you know, the codon usage is redundant. We have 64 codons, but you only have just over 20 amino acids. So it was difficult to go from protein to gene. But at the end, we managed, and we could clone the gene. And you see here that the gene is extremely small. It's a secreted protein, it's processed, and it ends up at a mature protein of only 28 amino acids. And all these cysteines, you see there, form disulfur bridges, so it's a very stable protein, and you see the structure on the right side with all the disulfur bridges. This took 7-8 years before we could do it. But those were the days that you could only find a sequence of your say ACTG by equipment that only could run for 20 or so base pairs. Okay, later on the job was easy, of course, once you've done it. So the next protein we isolated was AVR4, then AVR4E, then AVR4-9, and AVR5. And you see that all these proteins are very small. AVR2 is 6 kD, AVR9 is 3, and the biggest one is 11. They all have cysteine residues. And they all are secreted. As you see here, the same in the apoplast on top. The next protein, so the conclusion here also was, you remember, we discovered the proteins by their capacity to induce a hypersensitive response. So we knew they would induce resistance. But of course, a fungus, it would not be clever for fungus to make a protein to induce resistance. So there must be another function. We discovered, as together with others, of course, that many of these proteins had a virulent function. So their first function is to make the plant sick. They contribute to virulence. And so AVR2 does inhibit proteases, like here RCR3. You see in the apoplast, it's secreted by the fungus in purple. And then it binds to RCR3, which is a cysteine protease. And of course, that is a protease that normally would attack the fungus. Now it binds to the protease, it inhibits it, and so the fungus can grow happily. But of course, plants are clever. They have developed receptors to recognize the protein and then induce a response. What is nice of this protein is, and of this resistant gene, that it's also recognized by effectors from nematodes. So a nematode, Globoidea rostoviensis VEP1, is an effector that also is recognized by CF2. So plants are clever. They have receptors that recognize effectors from different pathogens, being a fungus or a nematode. The AVR4 effect is actually the one that occurs in many fungi. It's a chitin-binding protein. So you can imagine, if you're a fungus, let's say I'm a fungus here, and I'm covered with chitin, the chitinase of the plant would attack me, and I will be dissolved. So what the fungus does, it covers itself up with AVR4, so the chitinase cannot get access to the chitin, and that's the way it's protected against chitinases of the plant. And that's AVR4. This is the chitin binding domain. You see the structure on the left, and on the right you see the brown balls that are the chitin binding AVR4. And the green part is the chitinase, so it cannot get access to it. So, yes, so AVR4 is both an avirulence protein because it's recognized by CF4, but in the absence of CF4, it protects the fungus against chitinases. What then was discovered that AVR4 is actually present in many fungi. So this is a very universal protein that occurs in dotimycete fungi, and that consists of over 3,000 species. Most of them have these AVR4 homologs, which you see on the left. They all have this cysteine spacing. They all bind to chitin. And in the middle of this slide, you see it can bind to chitin. You can either use crab shell chitin or chitin from other sources. Chitin is insoluble. So if you put AVR4 to it, then AVR4 will bind and will go to the pellet, as you see in the two left lanes. If you add just cellulose, it will not bind, and then AVR4 stays in the solution. On the right side, you see that by adding AVR4 to mycelium, and then you add chitinase, it's protected when you add AVR4, and that's what you see on the two lower lanes on the right side, in this case with trichoderma. So both AVR4 of Cladosporium fulvum, of Microsphaera fijiensis, or of Septoria or other fungi, they all have AVR4 and they all are protected by AVR4 against chitinases. So now we're coming to Darwin and evolution and co-evolution. So how do fungi adapt to plants? So if the selection pressure is put on the fungus, of course, by mutation, will make a mutant that no longer is recognized. But how does it happen? With AVR9, it's very simple. It just loses the gene. But you would say that's not very clever, because by losing the gene, you also will lose its virulent function. But if you have 50 effectors and you lose one effector, you still are fairly virulent. So that's one way of escaping it. AVR4 makes mutants in the protein, so it has amino acid changes. Avia2 the same. And the same is for Avia4E. I mean, in some cases, you get a transposon insertion, so the protein is no longer expressed. So coming back to the Floor's gene-for-gene hypothesis and the quadratic check you see on the bottom, is by the mutations, you go from avirulence to virulence. What you see on the bottom, you have the leaf that is resistant, and then when the fungus makes a mutation or gets a mutation, it can overcome and avoid resistance, and then the plant is susceptible again. On the right side, you see why this happens. All these genes are located in parts of the genomes that are very repeat-rich, that contains transposons, retrotransposons, all these types that instabilize the chromosome, the genome. And this is the place when new factors are born and where factors disappear. And it's not only for this fungus, but many fungi do have this property. So factors are usually in this area. Okay, now we're coming to the receptors. So on the plants, I've talked about these effectors, and now we have the receptors in the plant. Most of those were discovered and described by Jonathan Jones, a colleague. We collaborated quite a long time. He works at the John Innes at the St. Louis laboratory. One of the genes was cloned also in the Netherlands, but the conclusion is that those effectors you see on the right side are located in the membrane. They are leucine-rich repeat. They have many leucine-rich repeats, but they have no signaling domain. It's like I give you a hand, the next one cannot give a hand to the third one because there's no signal telling what to do. So there must be something in addition. But I don't have time to explain that all, but I give you one slide where you see on top a few receptors. And one of those receptors is CF4. But in addition, you have co-receptors that are kinases. And kinases is a protein that if you phosphorylate it, it becomes activated and gives a signal to the next one. And so that, like SOBI1 and BAC1 and BAC3, these are very universal receptor kinases that work against bacteria, against fungi, against nematode, even against insects. They all bring down the signal from the outside to the inside of the cell, then activate the defense genes, and then the plant becomes resistant. What is interesting of this receptor is as soon as it is activated by an effector, what you see on the bottom side, the receptor is internalized in the cell. And then it regenerates and comes on top again. Because it's like if you have fever only for a few days. Because if you have fever, if you have a disease, you have fever for 10 days, you will be killed. So in order not to activate the plant all the time, these things are internalized, so then the plant knows how to respond, and that's good enough, not more. Okay, now I give you a very uniform model to understand the pathogenic fungus interaction, in this case as an example, Cladosporium fulvum. So these compounds you see on the bottom were the compounds we called in the 80s elicitors. These are chitin, glycopeptides, glucans, but now we call them PAMs. That stands for pathogen-associated molecular patterns. That tells a host plant, hey, there's a fungus. Or when there's a bacteria, hey, there's flagellin. I have a bacterium outside. I have to respond. In response, we call them PAM-triggered immunity. You see, the level of immunity is the dotted line. If below that line, plants are susceptible. If the defense is on top of that line, they become resistant. We call that PAM-triggered immunity. That immunity is turned on by those receptors. Okay, this explains why 99% of all plants are resistant to microbes. The clever fungi that are plant pathogens, they produce effectors, the blue dots, and those effectors suppress defense. They bring defense down to the level, the threshold, that normally gives immunity. No plants become susceptible, as you see on the slide. But plants are clever too. They develop new receptors, and they recognize these effectors. And that's what you see in the next. So AVR2, the effector I discussed with you, AVR4, ESP6 I didn't discuss, I didn't have time, but they are now recognized by another type of receptors. They are called the CF genes. And this is called effector-triggered immunity. That's a stronger type of resistance, and that's what several of you would call vertical resistance. While the first level is lower, many in the past would call horizontal resistance. Very small effects. Okay, now one slide I showed, actually some of you maybe have read it in the annual review of plant pathology, phytopathology. On the left side I show you, it's a summary of Cladosporium fulvum and tomato. On the left side you see that a PAMP like chitin is recognized by a chitin receptor. Then you get PAMP-triggered immunity. Well, the fungus is also clever. He produces a compound that prevents recognition of chitin. And then you get effector-triggered susceptibility. Well, then you get other compounds and they're recognized. And so this is what we call the rat race between host and pathogen. And so we have, with Phytophthora infestans, for example, there are over 20 resistant genes. There are over 30 effectors. And you would see if you not combine all these resistant genes in one or a few plants, plants will become susceptible. But if you can pyramide them, then of course you have resistant plants. On the right side you see, I just discussed with you Cladosporium fulvum, that's the right bottom. On the right left top you see the bacteria. On the left bottom you see, for example, mildew. On the right side you see pathogens like rust or magna pota. So all those fungi, bacteria, nematodes, they all produce effectors. They all are recognized by PAMPs. So there's a lot of interaction, but the rat race is more or less the same. So why was my talk called effector-assisted disease-resistant breeding? Actually, by using those effectors, we can inject them in plants, in wild tomato species, and then we look for hypersensitive response-inducing lines. And those lines can be used in disease-resistant breeding. So many of those we have against Cladosporium, but also many against Phytophthora in potato and other crops. So many crops can be protected by using natural defense, and those are resistant genes. We discover them by purifying the proteins, but some other groups, some of the colleagues in another department have used agroinfiltration, that is putting your gene or your potential effector into agrobacterium, PVX and agrobacterium, and then you can screen and look for HR-inducing plants. Well, here's the summary. So I talked with you about effector-triggered immunity. So plants have pattern recognition receptors, and that allows them to recognize PAMPs. Then, I know I have to look here because I cannot read it here on the screen, sorry. So with plant species, just want to be sure that well, to give a summary, so you have pattern recognition receptors, you have effectors, you can get resistant genes that recognize effectors, and that is the rat race going on. But actually what I was trying to say here also, in addition to what I told you already, that breeders, during the breeding process, have lost quite a few of the pattern recognition receptors. Because compare tomato in the wild in South America, they don't look like tomatoes, they look like ugly plants with small berries. But of course they're very resistant to pathogens. But we like to have big tomatoes that are delicious, that have many lycopenes, that all types of goodies. But of course, they've lost quite a few of those pattern recognition receptors. So that means if a plant with only a few resistant genes and having lost a few of these pattern recognition receptors, when it becomes susceptible, it's really 100% susceptible. So we have to be sure that we keep important pattern recognition receptors in plants. And that's what we in the past called horizontal resistance. On top of that, we have these strong genes. And we should try to combine them. It's unfortunate we cannot combine them at least in Europe by genetic modification, but now we have new tools to do it by genome editing. But also there the EU said last week that this will probably not be accepted. Well, that's about the end of my talk. Thank you very much.
Thank you, Pierre. And again, congratulations from all of us on your distinguished award. Welcome, everyone. The American Phytopathological Society, APS, is excited and very honored to organize and host this historical Congress. It's been 45 years since APS and ISPP came together to host an international Congress, and we're so pleased to do it again here in Boston. Boston is a city rich in history, known for innovation, entrepreneurship, and as an international center of higher education, which is a very fitting location for all of us to come together. On behalf of APS, I would like to extend our warmest welcome to you and our appreciation for your participation in this history-making Congress. Our opening plenary speaker for ICPP 2018 is Dr. Francisco Reifschneider. He is a Brazilian national who graduated from the University of Brasilia and received his PhD from the University of Wisconsin in Madison. Currently, he's a senior researcher at EMBRAPA and was former advisor to the president in charge of strategies. His ongoing activities are linked with international cooperation, coordinating the agricultural innovation marketplace and capsicum pepper breeding. During his presentation this afternoon, he will share his insight garnered from years of experience in international development and agricultural research. It's a real pleasure today to honor Dr. Reifschneider.
Thank you very much, and good afternoon. I would like, first of all, to congratulate Greg Johnson, Rick Bennett, Thomas Evans, Mary Palm, and their teams for organizing this wonderful event. It is a major task, and I'm sure we all praise and thank you for this effort. Let me also thank Rick Bennett, an old-time friend from still USDA years, president of ICPP, for having invited me to share with you some thoughts. It is an honor to be here. It is a privilege and a very special day because exactly 40 years ago, I attended my first ICPP in 1978 in Munich. I heard someone say that I must be 41 years old then. I think since we're talking about history, we have seen tremendous evolution in how we handle plant health. And if I were to pick an area that has made very significant contributions, all have, I would say that breeding and breeding for disease resistance has been of utmost importance to help us feed the billions of people that we have in this world. I think that today we are seeing a fantastic evolution in terms of new technologies, using drones, using sensors that are able to help us better identify where issues are. We are seeing different uses of mobile phones, such as the identification of a diseased cassava plant in Africa. Tremendous amount of new work coming, tremendous possibilities opening up for us all, I would say, to benefit from. But let me take a snapshot of today's world. And I think that despite the fact that we had a very long and productive journey, okay, let me try that again. All right. The snapshot is one that has both red elements, alerts perhaps, and green elements. We do have 700 million people hungry every day. 700 million people. This is a major challenge that we still have on Earth. We have rising inequalities within countries and between countries that also need to be addressed. We have conflicts, tremendous movements of people, of families around the world. We have climatic shocks, climate change, unemployment, a gig economy in certain countries that do not provide the safety nets that are needed by especially the younger generation. And I think we have a pendulum, like Foucault's pendulum, that today is moving anti-globalization, protectionism, and agricultural subsidies. But I'm confident this will move back. We have political and economic uncertainties of all sorts. But I also think, and I am by nature an optimist, that we have many, many positive things going on for us all. First, we have a collective commitment on the Sustainable Development Goals. And that has been achieved at great cost, with many discussions, but we do have a collective commitment. Our global food prices have gone down in the past decades, and this has helped us feed especially the poor. World trade has increased, and of course, trade supports diversified food baskets as well. The revolutions that we have seen in genomics, in automation, in sensors, in artificial intelligence, the digital space created by ICT, which allows, for example, my contact with producers in Africa using WhatsApp at any time, is really fantastic, because the flow of knowledge is really what is and can make a difference. And this flow of knowledge is very much at the base of cooperation. I understand that we have a tremendous opportunity yet to be harnessed in transdisciplinary research. Merging expertise fosters innovation. And this is something that we haven't yet touched with all the impetus, perhaps, that we should. We have new forms of financing which are also helping developing and industrialized countries move forward with innovation, with science, with technology. All in all, I think we have a fantastic possibility for the future, because we depend basically on global talents. And we have at least some 2,000 plus global talents in this room. We have global talents to help us move forward in our humanity. I'm sure you can individually add many different elements to the green list, and it's an interesting exercise to be done. And when we consider all this world, we must consider, of course, what is happening in terms of the world population. And I would say that it's a kind of a musical chairs game with one difference: nobody can be left behind. So if you look at what is expected to happen in terms of world population for the next 30 or 80 years, India is going to have the largest population. The second is going to be China. The third is most likely to be Nigeria. The expectations are for us to reach something like 700 million individuals in Nigeria, followed by the US, Congo, Pakistan, etc. The increase of some 2 billion or so by 2050 will pose tremendous pressure on the natural resource base that we have, already under stress. And of course, with all these changes that we see here, I think it would be fair to say that new political scenarios are and will be emerging. Now, this world population has an interesting characteristic. It has been urbanizing at a very, very fast pace. The lower map is 1970, and the yellow areas is where you had basically rural populations, less than 50% urbanized. Look at the upper map, what we expect to have by 2030. We are basically talking about an urban society. We are talking about less and less people producing food, and we are also talking about drastic changes in concentration. The circles represent very large cities, those with 1 to 5 million, up to 10 or more million, the red dots. Tremendous concentration. Yet, as I mentioned before, and I would stress again, the world poverty is something that should really be of concern to us all. We have some 700 million people living in extreme poverty today. That's about 8% of the world population. And although we should be happy seeing that thousands are escaping poverty as we speak, and a lot of that is very much associated with the work done by plant pathologists, by plant health experts, by agriculturalists, we unfortunately also see a few thousand falling into poverty today. And that we should try to avoid. The numbers are huge, and these numbers have to be considered ethically by all of us. We need, at the end of the day, to feed this population. We need to feed the world. And I used these two pictures just to compare a happy African family with a diversified diet and a happy European, Northern European family. But look at the difference in the food systems and the radical changes that we are all seeing. We know that beef demand is rising very fast in China and sub-Saharan Africa. And beef is plants. It is plant-based. Calorie intake is rising dramatically in developing countries in terms of cereals, roots, pulses, oils, and so on. We know that agricultural productivity must go by 60% by 2050 if we were to keep 9 billion people fed and healthy. 60%. Now, 2050 is not that far away. From the previous slide, we saw that Africa and Asia will contribute the most with this populational growth. And of course, we need to be concerned with food security because it's not just availability. It's not a mathematical, simple formula dividing what we produce by the number of people. It is about access. It is about utilization. It is about the stability of supply. We need to increase yields, indeed. We need to improve our production methods. We need to reduce waste and losses. But if you think a second, what is really different between these two families is the asymmetry of risks, the difference that they have because of different safety networks. Now, we as plant pathologists love graphs. I still do. And so maybe just a simple graph to illustrate the huge efforts that will be required to feed these 9 billion people by 2050. So if we just look at meat, the very first histogram to the left, or the very last cereals, we are talking about 50% increase in terms of cereals. 50% increases. And we are talking about 30 years. So, how are we going to do this? Are we going to be able to feed the planet with one agriculture? Is this the type of agriculture that's going to feed the world? This photograph was taken last year. It's an Indian farmer from extreme northern Brazil, completely isolated, zero external input, but he's producing peppers, he's producing cassava, and he's feeding his family. Or is it going to be this medium-sized farmer in the savannas of Brazil? The mid-size is just a joke. Producing a little bit of soybean. We need to produce this huge amount of food, of fiber, of wood, of feed, of fuel with plants. Is it going to be vertical, urban agriculture, as we have right here in Boston or in San Francisco? Well, I think not. I think it's going to be everything. It will continue to be a mixture from subsistence to controlled environment agriculture, CEA. And the reason for that, I think, is because we have a very, very diversified set of consumers. And our consumers, including myself, we come in all different shapes and forms and different expectations. What we as a scientific society need to do, in my view, is to adequately inform consumers so they may make the best choices they can. And to adequately inform, we need always top quality science and scientific integrity. Top quality science and scientific integrity. Our consumers, some love GMOs, but some are willing to pay $70 for a GMO detox kit. We have to respect our informed consumers, hoping that they will make the best possible choices. So I was delighted to see downstairs when I registered that 2020 is probably going to be the International Year for Plant Health. I hope we can all together work and help ICPP so that we can move in that direction, which will give us an added opportunity to adequately inform consumers. So we have a growing population, we have growing demands in terms of quantity and quality, we have all this demand to be probably satisfied by different forms of agriculture. We have demanding consumers, and we need healthy plants. So plant health continues to be central to this equation. And I decided to use Mr. Google to look at plant health and whether this was being searched often or not. Was this relevant to society? Not to this society. To this one, I'm sure it is. Is it relevant to the society that is outside with their shorts and Bermuda enjoying this beautiful Boston city and weather? And the answer is yes, very much so. And the amount of clicks searching plant health and plant health related themes has been increasing, which is very good. And the clicks are coming from just about everywhere in this world. So I see that plant health has continuously evolved. It will be of interest and relevance to society as a whole. But I had to choose just a few examples to highlight some of the changes that I feel are very relevant. The first one is disease detection. And I think Martinelli and colleagues have made a fantastic job in this small graph showing traditional detection versus innovative methods. going from visual symptoms, use of microscope, serological assays, to spectroscopy-based methodologies, remote sensing technologies, and so on. Very short period of time, very, very significant changes. I particularly like this slide, which shows work being done by two colleagues, Chazen and Drury, perhaps they are here today, using drones with spectral imaging cameras to look at the effect of pests and disease on cranberry. Tremendous opportunities ahead of us, and I think it's an excellent example of these diminishing disciplinary barriers that we have used in the past several decades. The second example I would like to use is focusing on collaboration and collaborative platforms, and of course, innovating financing. And I would like to use the words of UK Minister Penny Mordaunt that I heard in January of this year, when she says, "No one country has all the answers today. We need to work across continents on bold, innovative solutions that harness the best of human ingenuity. We know we can deliver spectacular results when we pool our resources and expertise." I know we can. And I look forward to seeing discussions along these lines this week. Now, collaboration and collaborative platforms are complex, but they're possible. And I'd like to use Rust UG99 as an example, because it is an extremely important disease on a most widely grown crop. And we had a Rust Summit in 2005 with Borlaug, Rockefeller Foundation, and CIMMYT, one of the CGIS centers, discussing how we were going to move forward. 2008, an international consortium was formed, Borlaug Global Rust Initiative, several institutions participating, but what's most important, over 1,000 scientists, more than 100 institutions. And then we had different initiatives also adding to this tremendous effort in international cooperation. The Global Rust Reference Center was established in Denmark with support from the Danish government by CIMMYT ICAD and Aarhus University. Rust tracking was supported by Bill and Melinda Gates Foundation, the FID, UK, FAO, and USAID. And that allowed us to move at a faster pace in terms of marker-assisted selection, in terms of wheat atlas and following the disease. A more recent initiative, Rust Watch, in the European countries, and a very important and many times forgotten political support coming from the G20 Ag Ministers meeting in 2011 with the wheat initiative. Complex, yes, but doable. Another example that I would like to very briefly visit is the agricultural innovation marketplace, that although it's not fully devoted just to plant diseases, you may see under news on the right corner that the very top is fighting the soybean rust. And here is a partnership to enhance agricultural innovation and development that has put together scientists, researchers, individuals from Africa, Latin America, Caribbean, and Brazilian experts, so that we could move innovation and development at a faster pace. This has been considered a model for South-South cooperation by the United Nations, but perhaps most important are the results that we have seen in a relatively short period of time. I'm talking about 10 years, 11 years. Thousands of experts trained, lots of knowledge generated, germplasm exchanged, products and technologies transferred. And look at the focus on the right, yield, a major focus on yield, followed by poverty alleviation associated technologies, NRM, natural resource management, and policies in general. And all this was possible because there was a large group of institutions supporting this collaborative effort. This brings me to a third example, the technological ecosystems that we begin to see and their financing. And the major change from a very complicated to a very complex architecture of today. Life used to be simple. We had either national or international, either public or private. Now, our membrane has increased its porosity between these four different... Oh, something went wrong here. Yeah. Increased the porosity between these four different rectangles. And what we have seen is an increased complexity. Why? Because private participation has increased and will increase dramatically. Limited public resources are available, both in industrialized as well as in developing countries. And private-public partnerships of all types have been generated with these new technological ecosystems. Now, the issue here, and this is an area in orange where usually plant pathologists do not like to dwell. You need to have adequate policies, you need right governance, you need checks and balances to avoid, for example, corruption, you need the right institutions and the technical capacity in the public sector. And the reason you need this is because you need to protect public interest at the end of the day. You need to protect public interest. A lot has been written about and published on public-private partnerships recently in all parts of the world. And perhaps this piece by OECD is one of the best I have seen. And the complicated to the left has been changed to a very, very complex architecture. These different ecosystems that we now have. Now, I wanted to use one very last example on the agricultural innovation hubs, because I do see that this is going to be an area where perhaps a lot of the younger plant pathologists and plant health experts in these rooms will find a space in the near future. The agricultural innovation hubs. And basically, if I use the AgTech Valley, it's one of the agricultural innovation hubs in a developing country, in my case, in this case in Brazil, my own country. It is the largest ag startup ecosystem that we have. And the characteristics are, you have an anchor company, a major company. You have venture capital coming from different parts of the country or the world. Academia, startups with entrepreneurs, accelerators, incubators. And perhaps, just perhaps, what I feel is still missing is the participation of the simple citizen. And I'm delighted to see that one of these days we're going to have, during ICPP 2018, a discussion on increased citizen participation. I think it is very important. Now, when I looked at more closely at these different startups and what was behind them, I thought it was very interesting to see that we had laboratories being developed for analytical work, precision agriculture in terms of irrigation, in terms of application of agrochemicals, use of drones, etc., etc., and everything concentrated around a city and therefore forming a hub. Now, what was not obvious was that plant health was at the core of all these activities, of all these new startups. And I found it fascinating to see that several of these new challenges that we are dealing with are beginning to be addressed by different systems of organization that we had before. Again, the porosity of our membrane has been changing all this. And I see as our biggest challenge exactly the speed of change. A tremendous change, not only in the technology, in the science, but in how we do business and how we are moving forward. And I think our new plant health expert has to move a little bit faster than Mr. Bolt. in order for us to really go ahead and do something which is a little more difficult than learning, unlearning. We are all used to the learning curve, and we are struggling with the unlearning curves. I think we have a tremendous opportunity here, because disruptive innovation is going to be more and more frequent. And you, the younger generation, will be dealing with disruptive innovation on a daily basis, from my perspective. And I hope I'm wrong, so we can have lots of discussions during the week. Now, despite all these tremendous advances that we have had, progress everywhere, our challenges are enormous. And I used just one simple photo as a reminder to all of us of the issues that we have to solve. And this is a huge processing tomato field, 2018, 100% loss. And you see Pierre is looking to the other side because he doesn't really want to see what has continued to happen here. Now, let me end with an optimistic message, especially to the newer generation. The same kind of message, perhaps, that I received some 40 years ago when I attended ICPP in Germany, in Munich. And it is about the Plant Health Expert version 2030. And I see this as an expert that needs to support this global movement towards a more sustainable future. One that has a strong desire to collaborate in problem solving, nationally and internationally. One that will be able to link disciplines and break these barriers, working with drones or mobiles or big data or tractors without drivers, big agricultural schemes or very small agricultural schemes, and who will have perhaps indeed many, many tabs open. But this is the new world, and one that will probably, most probably, have to venture into startups. I think that the need to work with different forms of agriculture, different forms of consumers, different needs, different expectations is at the base of what we're going to be doing for the next 20 to 30 years. The opportunities are and will continue to be enormous. And it is because of that that I feel that we indeed will see a very healthy future for plant health. Thank you very much.
Thank you, Francisco, for your enlightening presentation. It gives us a lot to think about during this week as we work to advance plant health. And I invite all of you to take him up on his offer to discuss the learning and unlearning and the disruptive science, etc. So thank you. An engaged world community of plant health scientists advancing knowledge for a safe, affordable, secure supply of food, feed, and fiber for a growing population. This is our vision for this Congress, and it's reflected in the wide variety of programming that you'll experience over the next 5 days. Today's presentations gave us both a historic look back at advances in molecular plant-microbe interaction research and a look to the future of plant health and the amazing opportunities that are before us, but only if we're willing and prepared to embrace change at a fast pace as our greatest challenge. So tomorrow we'll begin the day with the Monday plenary session, Plant Health is the Earth's Wealth. This session will feature 3 talks by speakers representing different areas of expertise and geography. Each talk has been prepared to set the stage for the Congress theme, Plant Health in a Global Economy. On Tuesday, the keynote session focuses on emerging plant diseases and global food security and features the Glenn Anderson lecture, as well as talks on climate change, metadata, modeling approaches, and the sweet potato as a food security crop. On Wednesday, we will learn all about new and exciting approaches to controlling insect-vectored plant diseases during Keynote Session 2. And those talks will cover utilization of effector targets, pathogen confusion to achieve disease control, and what we can learn from pathogen effectors. Reflecting our Congress theme, Thursday's Keynote Session 3 will give key insight into plant pathology's role in food safety. Presentations will cover mitigating aflatoxins, taking on the challenges of pesticide residues, and the risk management of foodborne pathogens. And we'll close out the 2018 Congress on Friday with the plenary session presentation entitled Global Food and Nutrition Security, From Challenges to Solutions. Bringing all of the ICPP 2018 programming full circle, this presentation will highlight the challenges we as plant pathologists face in addressing the growing problem of feeding the global problem of feeding a growing world population. And goals that we can set together this week, we can set for the next meeting, which will be held in 2023. And at that point, we can discuss how far we've come in meeting those goals. And now I'd like to welcome Tom Evans back to the stage to provide highlights about all the other incredible opportunities happening at this meeting.
Thank you, Mary. As I'm the only thing that stands between you and our opening celebration, I will be brief. In addition to these enlightening plenary and keynote sessions spoken about by Mary, the Congress offers a wide variety of programming such as panel discussions, phyto views, and a hot topic session on cacao. Scientific programming is not the only aspect of this Congress that will be of value to many of you. There are also a plethora of opportunities to share and network with colleagues while you spend your time here in Boston. During exhibit hours in the Veterans Memorial Auditorium, Exhibit Hall C, you will find buzzworthy idea cafes, inspiring phytopathologists of distinction we call pods, these pod talks, and an opportunity to meet one-on-one with an expert in your field. For students and early career professionals, there are socials and lunches to broaden your network and establish new relationships. Many fans are going to join together to celebrate and watch the Boston Red Sox game on Tuesday night for ICPP night at Fenway Park, and the truly American iconic House of Blues on Thursday evening for the closing will be an event to remember. If you haven't purchased your tickets yet for the closing event at House of Blues, please do that soon. For those of you that brought family along with you to the Congress, the family-friendly group has several activities planned for children at the meeting. Look for details on the bulletin board near the registration area. All of this programming can be found on the mobile app, and if you've not yet downloaded it, please do so. It's a wonderful tool to have on your mobile phone. And with that, I'm sure you're all ready for some socializing and networking. It's time for the welcome reception. We will all proceed together following our fantastic fife and drum players down to Exhibit Hall D. At that reception, you'll find an assortment of culinary options representing the local neighborhoods of Boston. Be sure you have your welcome reception ticket with you ready at the door to exchange for your beverage ticket. And thank you to everyone for your participation in this historic Congress, and we look forward to a wonderful week.